Good evening to all of you. And it's a real pleasure meeting you all again. This is the next lecture in the series of BioEdge seminar series. Initially, we started off with giving a very broad inputs on how this industry looking like, what are the sectors in this industry, and so on. And then we got into a conversation into what are the specific sectors, what are the specific industries within these sectors, within biotech sector, what are the industries, where are they located, what are the skill sets, and so on. And then we entered into a conversation on specific departments, each department, what they do, and what is the expectation in each department, and so on. Now, today, I would like to talk to you something very, very important. If you are a biotechnology learner, or you are a microbiology, or biochemistry, or pharmacy, or any of the life science students, any of the life science subject you are learning, and you are aspiring to get into industry, or at least if you would like to know how the industry works and what industry demands from you. uh, One of the key thing here in the industry is not about a great change to be made to become industry ready. Becoming industry ready is more of a mindset. Before I continue in this BioEdge Edge series, could any one of you from the Biocon Academy staff, could you please unmute yourself and say if I am audible as well as the slide is visible?
Sir, we can clearly hear you, and the first slide, Biocon Academy logo is seen.
Wonderful. Thank you very much. Yeah, if you are seeing that, we are absolutely in the right track. Wonderful. So that's what the point here is, to see how the industry readiness is looked at. Is it the readiness to work in multiple shifts? Is that called as industry readiness? Or hello? Okay. So basically, industry readiness is more of a mindset rather than transforming into something else. For example, let's say I'll show you one picture, and from there, I would like to start the conversation. Please let me know if you see the picture. Yes. So, if you are seeing this picture, it is not something very new sight. It is not something a very new thing what you are seeing. It is just a woman who is cooking something. When I say woman, with very hard heart, I'm saying that because it can be a man, it can be a woman, it can be your mother, it can be a sister, it can be your brother, it can be a father, it can be you, and it can be me. So basically, we are talking about a person who is cooking. When you see this picture, what are the things that are appealing to you? Whenever I speak about the industry readiness, whenever you are thinking about industry readiness, if you keenly look into this picture, you will see lot of things. Okay, for instance, you will see the raw materials that are required for cooking. You will see the materials that are required for cooking. You are seeing the environment that is required for cooking. You are seeing a person who is required for trained and required for cooking. The person is PPE'd, which means the person is wearing a personal protective equipment. She is wearing the appropriate gown. And you have a finished product in the other vessel. So many things you are seeing. For instance, I see the raw material, I see the people, I see the process. The process includes the method, the cooking method, and the utensils used for cooking, and the finished product, and particularly the environment. So, when you say cooking, cooking is the conversion of the raw materials to a edible finished product by people employing processes in an environment. If you carefully see, there is no harm in using this sentence for any manufacturing. It can be cooking, it can be dairy product, it can be medicinal product, or it can be anything. Again, I repeat so that we get this concept very clear. So that this entire conversation is going to be around this understanding. Conversion of the finished product, conversion of raw material to finished product by people employing processes in an environment. The process includes the methods and materials used, the methodology used as well as the materials that are used. In industry, you call it as equipment. In home, you call it as utensils and so on, right? So that is manufacturing. So what she is doing is a manufacturing of a food. When she is manufacturing food, what is more important? You might have seen that before serving this food to you, your mom will take a small scoop from the food and taste it. She's not actually consuming. The idea of that is not to consume food. The idea is to taste, which means she is doing a quality control before the food is served in onto the table, so that it is safe enough, it is good enough to be consumed, and so on. Out of all these components like people, finished products, process, raw material, if you consider this entire thing, entire manufacturing from the context of medicinal product, the most important one is the environment. The environment in which the medicinal product is manufactured. This environment is functionally classified into three major sets. There are three major ways we need to understand the environment in which the product is manufactured. The manufacturing is happening. The first one is called as built. When I say as built, as per the engineering drawing, a room is built. After the room is built as per the engineering specification, the room is equipped with equipment. The room is furnished with equipment. When the room is furnished with equipment which is not operational, then the room is called as at rest room. The room is furnished with an equipment, and there is an operator who is operating. Then it is called as in operation room. If you want to really understand from the industry's point of view how it is working, one of the most important understanding is to understand the industry environment itself. When I say industry environment, specifically I am talking about biopharmaceutical industry. Can be pharma, but it can be pharma and biopharma, not other industries. Specifically, I'm talking about these two industries in this context. So the reason why you need that type of importance to the environment is because the quality of the product is primarily determined by the environment in which it is manufactured. So generally the room is classified at rest or in operation. Can all of you mute the microphones, please? Satish, if you are there in the session, could you please mute the person? Okay, fine. So that's the importance of room classification. We will come to the designation of room classification little later. Before that, I would like to show you a picture where how the air is generated in the HVAC system, which is HVAC is heating, ventilation, air conditioning system, is supplied into the room and taken away from the room. As you can see here, the room is extremely well furnished in such a way that the air from the air generation system is supplied into the room and it is taken out from the room carefully. The cycle starting from the air generation system to the room and back to the air generation system, you call it as one air change. If I increase the number of air changes per hour, then the room becomes cleaner and cleaner. If you would like to understand this concept, it is easy to understand this way. The cleanliness or the quality of the room is dependent on the cleanliness and quality of the air. The air quality is dependent on 5 parameters. Number one, non-viable particle count, viable particle count, humidity, temperature, differential pressure. These are the 5 specifications which makes a room a clean room, a normal room a clean room. The difference between a classroom and a clean room is that in a classroom, you cannot do certain things which are done at the clean room, which means you have to do designated work in the designated area. That is why there is a classification of the rooms. As you can see here, there is an international classification, US classification, and European classification. International classification is ISO. For ease of work, I'll take okay. If you will able to see my cursor moving, ISO is the international classification. ISO 5, ISO 6, ISO 7, ISO 8. ISO 8 is the cleanest. ISO 5 is the cleanest, and ISO 8 is relatively less clean when compared to ISO 5. The same way, class 100, class 1000, class 10,000. The word class 100,000 should be used because you are talking about the US classification. United States do not understand the term lakh. They understand either million or thousand. So essentially, we have to say it is 100,000 grade, class 100,000. Europeans do not understand this classification. European way of classification is grade A, grade B, grade C, grade D. If you carefully see, the number of air changes per hour is the one that determines the cleanliness of the air. I already told you the cleanliness of the air depends on 5 parameters. Non-viable particle count, that is the suspended particles. Viable particle count, that is the bacteria and fungal count. Temperature of the room, humidity of the room, that is relative humidity, RH, relative humidity of the room, and the differential pressure. If you ask me what is the function of differential pressure, I'm sure most of the people who are attending this session would have gone into any of the religious places. It can be a temple, or it can be a church, or it can be a mosque. You might have seen as soon as you enter into the religious structure, you will see the pressure inside the structure is more when compared to the pressure outside, which means the air from inside can go out, the air from outside cannot come into the room. This is called as the differential pressure, which means wherever you want cleanliness, you create a positive pressure in the room in such a way the room air can go out, the external air cannot come into the room. This is achieved by increasing or decreasing or having the air change per hour in this system. Up to here, we have spent a lot of time in understanding what is the environment in the pharma biopharma industry, which are called as the classified rooms. In classified rooms, there are only two types: classified uncontrolled rooms and classified control rooms. In a classified uncontrolled rooms, there is no air changes per hour control. There is a air cycling, but there is no control over it. But in a classified control room, then you can call it as class 100, class 1000, class 10,000, and class 100,000 based on what you want to do in that room. For example, if I take the manufacturing of a medicine, the manufacture of the medicine in a typical biopharmaceutical industry starts with fermentation. The fermentation is the upstream process. After the fermentation, the product is recovered, purified, concentrated, formulated, sterilized, and filled. These are the processes. You can easily imagine the purity of the product will be increasing over the downstream. The purity of the product increases over the downstream, which means the downstream operations should be carried out in a cleaner room when compared to the upstream, because there is a direct correlation between the room cleanliness and the purity of the product. More the product is pure, more the pure the air in the room is. That is how it should be understood. So now it becomes easy for all of you to follow this classification of class 100, class 1000, 10,000, and 100,000. If I take an example, in class 10,000, I can do fermentation. In class 1000, I can do the downstreaming. When I say downstreaming, I can do the recovery and purification and concentration and so on. In class 1000, probably I can do the formulation. In class 100, I'll do the filling and closing. So if you can see in this entire unit operation wise, there is a clear correlation between the product purity and the air cleanliness. That is the importance of the air and air quality in the biopharma industry. This being said, we'll go into the next extent of understanding. Give me only 1 second, something wrong here. Wait. Okay. So now we'll see again, revisit the sentence which we used to describe the manufacturing process. Conversion of raw material to finished product by people employing processes in an environment. That's what we spoke about so far from the environment side. Now I would like to spend some time in terms of giving a little more information around the good manufacturing practices. When you say good manufacturing practices or GMP, the good manufacturing practices are not restricted only to the manufacturing facility. As you can imagine, the product when it is manufactured, if you go back to the same example, the food that is getting manufactured in the kitchen is tested in the kitchen, which means that the product that is manufactured in the manufacturing room has to be tested in a facility with the similar cleanliness of the air. That means quality control also is governed by good manufacturing practice from the environment perspective, people perspective, process perspective, because they are the ones who are going to test the raw material also, finished product also, in process material also. So the GMP has a larger scope. It is having a scope all the way from the development of the product, manufacturing of the product, quality control of the product, storage of the product, and release of the product into the market. In this case, what is the most important objective of good manufacturing practice? If I have to rephrase this question from this context itself, what is the most important objective of a pharma company to manufacture the product? Manufacturing the product is an objective, no doubts about it. What is the importance or what is the objective of manufacturing the product? The objective of manufacturing the product is to give the best patient experience. For any biopharma industry, the objective of manufacturing a drug product, medicinal drug product, is to create an excellent patient outcome, patient experience. When I say patient experience, it has two elements to it. One element is called as potency, the other element is called as safety. For example, in one of the launch of the program that has happened recently at Biocon Academy, my colleague Susan Alexander, she used a very nice analogy. She said, when you are taking a pill, for example, you are taking a pain medication or you are taking a fever medication, when you are popping up a pill, you are not actually thinking whether it will work or not. You are not wondering, you are not testing it out. You are not thinking that let me try this out. You are believing and taking it. You are believing and consuming it. You are believing in two things. One, the medicine which you are taking, it will serve for its purpose. That is your first belief. Your second belief is that it is not going to create any harm to you. One is it is going to alleviate the disease. One, it is going to treat the disease. At the same time, it is not going to create any harmful effect in you. Which means a patient experience has two major objectives. One is the relief and other one is the safety. If from a technical point of view, you can keep it as potency and safety. Potency means effectiveness, efficacy, efficacy of the product, efficacy of the API, the actual medicine, and safety, how safe it is. Is the product creating the known side effects? I don't have to tell you this fact that there is no product that doesn't have a side effect. If you have the habit of reading the pack insert that is given with the product, with the medicine, for example, if you buy a tablet or if you buy a capsule or if you buy a ointment or oral suspension or injectable, you will see a small pack insert that is inside the box. If you open that and see, it will be a little hard to read because it is in font number 6 or 7. But actually, if you spend some time in reading it, you will see that it is clearly mentioned for what are the conditions this product can be used, under what conditions it should not be used, what are the known side effects it will create, what are the side effects it will create if it is abused. That is the clinical trial outcome. That is adverse drug reaction it will create if it is abused. So these are the information already given in the pack insert, which also means that this product is anyway going to create a side effect. Most of the times we don't even know what is the side effect that has come up unless it is very obvious. For example, if it is nausea, if it is skin rashes, then you will get to know. But if it is slight dizziness and all, you will not get to know because you are thinking that it is a part of your disease, right? So when you say the patient outcomes are safety and potency. The safety and the potency has to be built in a product through GMP. When you are using a good manufacturing practices to manufacture the product to build these two qualities in the medicinal product, the most important focus inside the industry is given not only to build the quality, but build the quality consistently, which means if one batch of the product is giving best uh, patient experience, excellent, very good, thumbs up. What if the next batch is not? What if the next batch is not giving as good as experience it has given in the first batch? That is not acceptable. Which means in the patient outcome, hello, in the patient experience, it is very important to create a consistency. So the objective of the good manufacturing is to produce consistently good quality products, or otherwise consistently good quality products which are meeting the predetermined quality expectations by the regulatory agencies. That is the objective of good manufacturing practice. When you say you want to create a consistency in the quality, what are the things you have to focus? You have to focus on reducing the variability in the raw material, reducing the variability that arises out of people, reducing the variability arises out of process so that the finished product doesn't have any variability. The measures that you are taking to reduce the variability in these components are collectively called as good manufacturing practices, of which only one component we have seen in detail, that is the environment. The second component is raw material. In the environment, before I go to environment, in environment, already we have seen this type of classification, international classification ISO, United States classification classes, and European classification grades. Now coming to the raw material. In a biopharmaceutical manufacturing, one of the very, very important raw material is the medium. By now, all of you know, if you are a microbiologist or a biotechnologist or any life science student, in industry, there are two major platforms in which the products are manufactured. One platform is a microbial platform, other platform is a cell culture platform. In microbial platform, you can use yeast or bacteria or whatever. And in case of mammalian cell culture, you can use a range of mammalian cell lines. For example, it could be CHO, Chinese hamster ovarian, or it can be Vero cell line, or it can be BHK21 cell line, and so on, depending on what you want to express. Okay, so basically there are expression systems. So based on the expression system, you will be using different types of media. If it is a microbiological expression system, if it is a fermentation, then you will be using microbiological media. If it is a cell culture, then you will be using minimal media. Therefore, media is one of the most important raw material that has to be controlled as a part of good manufacturing practice. How do we control? There are two ways. Number one, the media that is produced in-house. If you are producing the media in-house, which means in your own company, 2000 liters or 5000 liters, whatever may be the scale be, if you are manufacturing the media has to go through a test which is called as GPT, growth promotion test. You are going to inoculate the known cultures into the media to find out whether the media is capable of supporting the growth of the known organisms. So that is the GPT through which you can control the quality of media. The media itself is made of multiple components. If you're a microbiologist or studying microbiology, you will know that there is a yeast extract, beef extract, peptone, NaCl, and so on. If you are making the media by individual components, you have to have a vendor certification that these components are good quality components. So the validation can either be in-house or the validation can be through vendor audit. You can audit the vendor and see whether the vendor is producing and giving you the good quality media and media components. This is one way of understanding the media. The other way of understanding the media is the media preparation can be outsourced. You can outsource to a company which produces only media. Sometime back, we had a company called a Sigma Aldrich, uh, opposite to Biocon facility. They used to produce only media on an order basis. There are a lot of companies, and not supporting any particular company. There are a lot of companies who are into media manufacturing. It can be high media, Merck, and so on and so forth. Right. So basically, media can be outsourced. Media manufacturing can be outsourced. In that case, what you need to do is you have to only do the qualification of the vendor. That is the first portion. Second portion is the cell. Cell is one of the most important raw material because this is the one which is going to produce the product of interest using the media components. We told examples of E. coli or Saccharomyces cerevisiae, or it can be Serratia marcescens, or it can be Pichia pastoris, or it can be Aspergillus niger from the microbiological point of view, right? So these are the cells. When you say cells in the pharma biopharma industry, we call it as expression system. Why we call it as an expression system is because using recombinant DNA technology, we have inserted the gene of interest, which is going to produce the product of interest from the cell which you are using. If I have to take an example, let us take example of Pichia pastoris. A native Pichia pastoris doesn't have the ability to produce insulin. A recombinant one does, which means that a recombinant cell of Pichia pastoris is produced using rDNA technology by inserting the gene of insulin producing gene into the yeast so that it becomes a recombinant yeast. That is the cell line. Again, there are two ways. If you have the capability, your own R&D can do the clone development. Otherwise, you can outsource the clone development also. In that case, again, the same thing I told for media, you have to go and audit the facility which is developing the cell line for you or the clone for you. Once it is done, the cell has to go through a characterization step, which is called a cell line characterization. You have to prove that the cell is good enough to be used as the inoculum. Then comes water. Water is one of the most important component raw material in the pharma biopharma because it is not only used for in manufacturing the product in the formulation but also in all the other ways. For example, for entire cleaning of the equipment, stainless steel equipment, water is used. The raw water, tap water, groundwater is used for cleaning the facility. The purified water, PW, is used for media preparation as a solvent. The water for injection is used as the solvent for formulation. So different types of waters are there. And there are some cases in analytical lab, they'll be using a demineralized water or deionized water. Okay, so these are the different types of waters that are manufactured and used in the manufacturing facility. All these waters has to be individually qualified and validated for its activity. The next important one is the air and the gas. Here I'm not talking about the environmental air. We already spent time in understanding the environmental air, so I'm not going into details of that. I'm talking here, we are primarily talking about the air that is used to move the product from one vessel to other for all the pneumatically actuated ones. Harshita, do you have a question? Could you please,
Sorry to interrupt you, sir, but your voice is not clear.
how about now?
Hello?
How about now?
No, sir, not yet.
Sir, your voice
Your
is clear. I
voice is
think
very
some
clear.
network problem from her end.
Okay, because Harita, since it is only for you, maybe it is a case that it is a network issue from your end. Please look into it.
Okay.
Okay, okay. Because all the others they are able to listen. So the air, what we are talking about here is the air that is used in all pneumatically actuated systems. Pneumatically actuated system means all the instruments that are using air as the source to actuate their valves. Then it is called as a pneumatically actuated system. It is based on the air pressure. All the instruments that works based on the air pressure. And particularly some of the systems where air is also used as a raw material. For example, let's say in fermentation. When you're doing the fermentation in a large fermenter, uh, you have to maintain the dissolved oxygen. When you maintain the dissolved oxygen, there is a instrument called sparger. The sparger is connected to a sterile air inlet so that you can supply the air. When I say air, either it could be air or it could be gas. For example, in this case, it could be oxygen. You can supply the oxygen. The oxygen will go through a sterile filter, get sterilized, and go into the sparger, go through the sparger, and get sparged into the microbiological medium. Okay, so this is very important because this air or this gas is the one which is going to give a conducive environment, a favorable environment for the cells to multiply in the media. So manufacturing, generating the air or procuring the air, purifying the air, sterilizing the air, supplying the air is very important function and it is a part of the raw material. These are the four raw materials which are important if the company is manufacturing the API itself. When I say API, it is called as active pharmaceutical ingredient. In the industry, there are two major categories of companies. There are companies who will manufacture the API. When I say manufacture the API, the active pharmaceutical ingredients. If I take example of paracetamol 500 mg tablet, if I take paracetamol 500 mg tablet, this 500 mg paracetamol is called as API. All the other components like bulking agents, coloring agents, preservatives, stabilizing agents, all these are called as excipients. Mixing the excipients and the active pharmaceutical ingredient together is called as formulation and converting it to the finished product. So the first four components what we discussed so far are very important for those who are manufacturing API and the finished product. But there are companies who will buy the API directly and do only the formulation. In that case, API itself is the raw material. So in case of API validation, again, as I told you, you have to do the vendor qualification for people who are manufacturing the active pharmaceutical ingredient or bulk drug. With this, I would like to stop the raw material portion and we will move on into the people component. People component is very, very important component in good manufacturing practice because, as I told you, the objective of good manufacturing practice is to reduce the variability. As all of you can easily imagine, one of the important source of variability is us, we the human being. We are the ones, we will one way or the other create a variability. That's why we normally use the term called as operator subjectivity. It is not always the same. It depends on the operator, right? So basically what we need to do is we need to find out, we have to do a exercise which is called as gap analysis. In gap analysis, the HR and quality assurance, what they do is they try to understand what is the qualification you are coming from, what is the job expectation, and what are the skill gap you are having. They have found out that you are good enough for being an employee. Now, after becoming an employee, you have to get trained before you do the job function, before you start delivering the job function. For that, there has to be a gap analysis, and there has to be several training programs. Both technical and professional skills trainings has to be given so that you become a full-fledged employee to start delivering these job functions. One of the important training that happens is called as SOP training. SOP refers to Standard Operating Procedure. Standard Operating Procedure is one of the key document which is having the capability to reduce 90 to 95% of variability. If all the employees follow the follow the SOP, majority of variations can be controlled, can be reduced. It has that type of power because SOP is written after all the procedures are validated, standardized. That's why it is called as standard operating procedure. That training is given to all the people. In addition to that, the health of the people and hygiene of the people are continuously monitored based on whether you are a critical resource or you are an enabling resource. Critical resource means you are directly involved in the manufacturing process. Enabling functions are the supporting functions to be for the company to be successful in implementing the good manufacturing practices. That's the people component. Coming to little strong and complex GMP component is the process component. As I told you, process is a combination of equipment and methods. That's the combination called as process. In the process, one of the key thing in the industry is that what is written is done. What is not written is not done. That is the power of documentation, which means whatever you are doing has to be a result of what is already being written in a document. For instance, you want to measure the pH of insulin. You have manufactured the insulin as a part of quality control. You want to measure the pH of insulin. You have a pH meter in front of you. You have gone through a pH meter training. You have gone through the SOP, how to operate the pH meter, which means that you are going to follow a SOP, written SOP, to test the product, which means to conduct the process, there is a document. After following the SOP, you are measuring the pH by following the SOP, and you are measuring the pH and documenting the pH. You are seeing 6.8 as the pH. You are writing this document. You are writing in your raw data sheet, RDS, that it is 6.8. Or in some sophisticated one, you will get a printout itself. Right? So that is the outcome, the document outcome, which means what is written is done, what is written, not written is not done, which means whatever you are doing should be a outcome of a document. Whatever you are documenting should be a outcome of process you have conducted following some document. That is the importance of documentation. That is the reason why generally a BMR is a large document. BMR is a very voluminous document. When you say voluminous document, it has a document on every stage from the starting of the vendor qualification, vendor audit, vendor qualification, media composition, cell characterization. Everything is a part of, and process parameters, monitoring of the process parameters. Everything is a part of BMR. BMR refers to batch manufacturing record. Every batch of the product has a record of manufacturing. This is very important because tomorrow, if there is a small error in this product, it can be backtracked correctly, accurately, because everything is documented. That's the power of documentation through BMR and trend analysis. One of the most important initiative the regulatory agencies came up with is called as IPQC. That is called as in-process quality control. Sometime back in the past, in the history, only the final product quality was important, which means that if the final product quality meets the predetermined quality criteria outlined by regulatory agency in the monograph of the pharmacopoeia, you can pass the product. But now it is not like that. You have to have, when I say you, the manufacturing company have to have in-house specification for in-process quality control. So in-process quality control is most important one. This is achieved by sampling and testing. In every stage of manufacturing, sample is collected, tested, the outcome is referred, outcome is checked vis-a-vis the in-house specification. If it is less than the specification, then you can move ahead with the next step of manufacturing, which means the in-process quality of the material is very important, as good as the importance given to the quality of the final product. That is about the in-process quality control, and the in-process quality control and the final product testing, irrespective of that, if you see, it is in the EMP, in water and product. EMP is Environmental Monitoring Program. In Environmental Monitoring Program, Quality Control Microbiology Department does the environmental monitoring program, where they find out the microbiological attribute of the air, the microbiological attribute of the people, the microbiological attribute of the surface, because these are the contributing factor for contamination. They also test the microbiological attributes of the water that is being used and also the product. As I told you, it is not only the finished product, but also the in-process product. So all these intermediates, all these are tested by the quality control microbiology department. This is why I told you GMP is not restricted to only manufacturing. The term manufacturing is a very encompassing term, which includes everything within the scope of the company. How they perform this activity, how they manufacture, how they perform the operation, how they actually execute the manufacturing, and how do they take the samples, how do they test the sample. Everything depends on SOP. This is what I was initially talking about when we were talking about training. Standard operating procedure is the one which is going to reduce the variability to a greatest extent in the biopharma industry. The standard operating procedure is originating from method development protocol, which means in the R&D, if it is a manufacturing process, there is an upstream development group and downstream development group. And if it is for quality control, there is an analytical method development and validation group. So these group develop the method for manufacturing and testing, and all these comes through the tech transfer document, method development protocol, to the specific departments, to the manufacturing, to the quality control, and so on, where it is converted to SOP and standard testing procedures. And then these SOPs are used for training of the people who are actually going to conduct this entire manufacturing. It is not only important to train the people in how to manufacture the product using the equipment, it is all the more important to qualify and validate the equipment itself, because the dependence on the equipment is extremely high. We cannot manually manufacture a large quantity of product. We have to depend on the equipment. In our house also, if you see, if four of you are there at home, you will use a small cooker. And if suddenly there is you are expecting a lot of guests, you'll use a larger vessels, right? So the variability is very high in the utensils and the equipment. The equipment has to be chosen appropriately as per the unit operation, and then it has to be qualified. The purpose of qualifying the equipment is to create dependency on the outcome the equipment is going to give. consistency of the outcome the equipment is going to give, for which the equipment is qualified for 3 stages: installation qualification, operation qualification, performance qualification, IQ, OQ, PQ. All three put together comes under a larger picture called as validation. The equipment has to be validated, which means the equipment has to be identified correctly. We call it as URS making and so on. So basically, based on the URS, you are getting the equipment. And once the user required specification gives the reference to the equipment, and then the equipment has to be installed properly. When I say installed properly, it is not random. You have to choose the place where it has to be installed. You have to give the proper power connection. You have to give the proper environment for the equipment to work properly. All together is called as installation qualification. And then operate the equipment. When you are operating the equipment, you have to see the equipment is having the ability to operate from the lowest range to the highest range. For example, if you're installing a centrifuge, you have to make sure that the centrifuge is able to run starting from 100 all the way to 10,000 or 15,000 RPM. This is called as the operational qualification, where there is no product involvement. The performance qualification is product involved. For example, if I take the same example of centrifuge, you want to get a pellet of yeast. Here you have to use the yeast solution, yeast culture, and find out at what particular RPM you are able to get the maximum pellet. This portion of using the equipment for the expected outcome is called as the performance qualification as a part of validation. This entire process is overseen by two set of people. One is your internal quality assurance people, the company's internal quality assurance people, through a process called as internal audit. They will do audits to find out if everything is going fine. The other way of understanding the audit is external audit. There are two types of external audit. If you remember, we said there is a media preparation company for like that. If you are a drug product manufacturing company for other company, for example, let's say X company is manufacturing for Y company. Y company is going to market it. In that case, the Y company will come to X company and do an audit. This is called as a vendor audit. For instance, we said you have to go, if I'm a drug manufacturer, I have to go to media company. For the media company, I am the vendor audit. I'm performing a vendor audit. That's also an external audit. The other type of external audit is the regulatory audit. If you want to sell the product in the US, then FDA will come and audit your facility. If you want to sell the product in the Europe, then EMA, European Medicinal Agency, will come and audit your facility. Like that, the external audit also will happen. The purpose of all these raw material qualification, people qualification, process qualification, equipment qualification is all to make sure that the final product which you are manufacturing is having good quality. When I say good quality, it is going to create the best patient outcome in terms of efficacy and safety. Here also, the quality control microbiology and quality control analytical department play a very, very important role. When you say efficacy, what they do is that they conduct quality control testing on the final product. For example, if you are familiar with the equipment called HPLC, high performance liquid chromatography. So if the HPLC is used to find out what is the concentration of the product in the final vial. They are taking vial from the final product and they test it out. This much of quality testing is done before the product reaches the patient for the final consumption. The quality control analytical people will test the assay. Assay means what is the concentration of API, what is the concentration of the excipients, what is the concentration of, most importantly, the impurities. All these are tested and find out whether everything is within the set specification. That is the safety testing, because the safety testing includes the impurity profiling. Microbiological point of view, if it is a sterile product, a sterility test is done to find out if there is any microbial contamination in the product. if it is a sterile product or it's a non-sterile product. The test is specifically done to find out if there is a biological impurity, like bacterial endotoxin is present or not. All these are the part of the safety and efficacy test to build quality in the product. To build the quality in the product consistently, to create quality in the product, the only way is to follow the SOP. SOP is one of the important component in the good manufacturing practices. I think I'll pause here and I would like to take any questions, if any. We have 10 more minutes to take questions. The chat. What type of filters are used in HVAC system, and what about their size? Oh, wow, excellent. Already there is an answer. Okay. How many times external internal audits is done in pharma biopharma companies? Whose role is to conduct these type of audits? I think we have already mentioned about internal audit and external audit. There is a specific frequency in which the internal audit is done. The frequency is also set by the QA department. As far as the external audits are concerned, it is completely on the need basis. Whenever FDA wants to audit a facility, they can audit by giving a prior notice, or sometimes if you want to sell the product as a manufacturer, if I want to sell the product in the United States of America, then you have to file your product through the regulatory affairs department, and then they will convene a facility audit by FDA. When I say FDA, the external audit agencies. High quality product to patient-centric approach for saving lives. Quality cooking skill that lady. Having cooking skills that lady inbuilt in her. I may beg to differ here. Actually, if you see, this is also a training. Her mom trains her. When there is a training, there are multiple stages in the training. Number one, the person who is getting trained is not directly allowed to do anything. They are supposed to watch the others do the process. The same way that lady would have been watching her mother doing things, right? And then it becomes a inculcated or inbuilt ability for her, because there is a reproducibility of the result and repeatability of the process. So it is not something inbuilt. It is not innate. Certainly, it is a training. We need to believe this, because then only we'll get to understand why we spend so much time in creating trained talent pool for the pharma biopharma industry. My sincere request is, I don't know who is this RN. Please do not record or document this session without a written permission from Biocon Academy. That is my sincere request, because coming from pharma biopharma industry perspective, this is considered as a data integrity breach. So if you are learning from the beginning, you learn to ask permission before doing something. What if I done my wrong sign on a control copy? Very simple, strike it. In a good documentation practice, what is expected is first time right. You're supposed to get things first time right? But at the same time, it allows you to correct it, because end of the day, you're a human being, right? So I think we are talking about Swapnil's question. Swapnil, if you have put a signature wrong, strike only once and put a signature next to it and put the date and move on. Every company has a specific way to do it. Only thing you are not supposed to do is overwrite. Just because you have done something wrong, don't enter into overwriting. Overwriting is not a good practice from industry's perspective. So deionized water and demineralized water both are same. No, not really. The deionized water and demineralized water depends on what is the equipment which you are using. For example, if you are using a multi-column distillation plant, then you are talking about demineralized water. And if you are using RO resin, then you are talking about deionized water. It depends on what is the purpose of the water which you are manufacturing. So it depends on that. So it is not the same. How many times extra that we have already answered? Are there any suggestions from your side to enhance the skills? Skill enhancement is a dedicated practice. I'm really happy that you are asking this question, which means you are in the road of focusing on it. That's all you have to do. Keep focusing on skill enhancement, spend time, keep some money for that skill development. You can do it. Regulatory audit and statutory audit will be the same. Yes, absolutely right. These two are the same. Conventional small molecule based pharma is less cost and has widespread use. What about the future of biopharma industry in India and widespread use of this bio? Wow, this is really a very strategic question. Yasmin, as you rightly mentioned, there is a difference between a small molecule and a large molecule, right? So it depends on a lot of things. It depends on the what is the affordability and what is the support system a person has to choose chemotherapy or a chemotherapy with a biologic. So it depends on that, right? If somebody is in the US, it is covered as a part of their security number. And if they're in India, if they have kind of support systems to do that. So it is based on that. So based on that, as you can see, there is a difference between a volume game and a value game. When you say game, I'm not talking about a game in its sense. What I'm saying is the model, value model and a volume model. Small molecule is a volume model. More you manufacture, more you get profited as a company, as a pharma company. Less you manufacture, but you are manufacturing a high value product. That is the value model. So both are sustainable. You cannot compare a pharma company and a biopharma company from the standpoint of its future. Each of it will have its own future in its own way. But I can't strike out. Why? If it is wrong, you have to. What if somebody says this is not your signature? You are not the person you have done the signature. Then that validity of the document is lost. If you are the creator of the document, you should have a mechanism to strike out whatever it is. Whatever it is written by using a pen should have the flexibility to be striken through. So if you are saying you cannot strike the sign, what is the point of having a wrong sign? So that's not the point, right? So that's not the good idea. How can we define analytical quality, leadership, acquired quality limits? See, the AQL is more of one of the parameters. It is one of the parameters in developing the quality system. AQL is not the only parameter. So AQL should be considered as what do you call approach. It is more of one of the approaches to ensure quality. Because we have one more minute, I would like to quickly move forward. In the upcoming world with AI and its advancement, how shall it make changes in the GMP? Will it replace human working in biopharma? I would like to close with this question. AI is a friend for you. He or she is not a competitor for you. AI is not a competitor for you, right? You have to start understanding how to collaborate with AI. Because you are the one who is creating the AI. So human power is the one who is creating the AI, which won't take away, which won't replace, which will much more enhance. Which means the people who are working in the biomanufacturing industry, if they know how to operate in the AI system, will sustain. Otherwise, the people who have that ability and skill set will replace that person. So it is not AI replacing. The AI skilled person is replacing. So it is not replaced by technology, it is replaced by another human being. All right. So with that, I really thank all of you. I'm seeing 106 participants. It's really very convincing to see a lot of people participating in the BioEdge series. And thank you so much for your support. This is really encouraging sign for all of us to continue to do that. Thank you very much. Have a good rest of the evening. Bye to all of you.
Thank you, sir, for the wonderful presentation.
No
It
problem.
was
Thank you very
absolutely
much.
nice. Thank you.
Thank you.
Thank you, sir.
You're welcome.
Thank you, sir.
You're welcome.
Sure, I had one doubt which I actually typed in the chat box. So if I want to write a new SOP, where can I refer? Like,
SOP has to be an outcome of the protocol. As I told you,
okay,
if you are writing a SOP, which means it is a operational nature. You are not writing a
okay,
SOP for a testing. If it is a operational procedure you are writing, it has to come from the upstream method development protocol or downstream method development protocol. You cannot straight away start writing SOP.
okay. Or if I want to improve an already like existing SOP, how can I?
Then it is a CAPA. The reason
Okay,
why you are improving your SOP because there is a deficiency.
yeah,
That deficiency you would have corrected through corrective action, preventive action. So from corrective
okay,
action, preventive action points, you have to take as a change control and write it
okay.
that way.
Thank you, sir.
No problem.
Hello, sir. I'm
Hi.
Arshita. Sir, I would like to know if there are any internships available at Biocon for freshers right now. If you
Harshita,
have any information, please share with us.
yes, I'll do one thing because it is out of context. Please connect me through LinkedIn and post this question in the message. I'll respond to it.
Okay, thank you so much, sir. Thank you
You're
so
welcome.
much.
Yes, Sonika.
So I had the same question as her.
Same answer for you also, Sonika.
Yes, sir. Thank you.
No problem. I'll meet you in the LinkedIn.
Sir, I have one more question also. Sir, I'm
Praveen,
a BTech biotechnology student.
okay.
Sir, I can like I know how to operate in reactor. But I
Okay.
feel like biotechnology business, like also BTech and BSc business are like taken for like quality control or in upstream. Like I feel like I didn't get an opportunity like in a production. Like chemical engineering students are like uh, highly getting opportunity in the production field. Is it true or like I feel like that?
Praveen, it is very, very untrue.
Oh,
The manufacturing quality control facilities are multidisciplinary. Everyone has their
okay,
own role to play. There are biochemists,
okay,
there are chemists, there are microbiologists, there are biotechnologists, there are pharmacists and chemical engineers. Everybody has their own role to play.
okay,
So you will definitely have a role to play.
okay. Thank you.
Sir,
Okay, I think it is time.
sir, I have,
I take this as a last question, Drishtiman.
thank you, sir. Sir, actually, I am a postgraduate in clinical research, in clinical trials. So, sir, how to get inside a biotech industry as a junior like trainee, or how to get in the biotech industry, sir?
if you are into clinical research, it is really a good idea to first start with those CROs, that is contract research organizations, who are conducting clinical trials for different products. You cannot specifically get into biotech directly, because you may not have a visibility whether the product that is being tested is a biotech product or not. So basically, your idea is to first get into a contract research organization. From there, you can find your way to a biotech industry.
Okay, sir. Thank you, sir.
You're welcome. Thank you, and I would like to log off. Bye to all of you.
Thank you, sir.